So the third and final panel will be on human genome editing, navigating ethics and global governance. Moderating this panel is Dr. Jeff Kahn, Dr. Andreas Dracopoulos, Director and Levy Professor of Bioethics and Public Policy at the Berman Institute of Bioethics at Johns Hopkins University. He was also one of the expert members of the planning committee, and we're really happy that he was part of the planning committee and that he's here today to moderate this panel. Please welcome Dr. Kahn and the panelists to the stage.
Thank you, Angela, and thanks everybody for coming back after the break. It's been a great day so far, but I know it's hard to come back as we get deeper into the afternoon. So as everybody gets settled, I will first introduce our panel and then provide just a very brief overview before we get into the discussion from our panelists. So they're not sitting in quite the order that I have listed here, so I'll ask you to raise your hands when I identify you. So speaking first will be Feng Zhang, who's there, who's the James and Patricia Poitras Professor of Neuroscience at MIT. He's also an investigator at the McGovern Institute for Brain Research at MIT and a core member of the Broad Institute of MIT and Harvard. Following Feng will be Pete Mills. Peter is the Assistant Director at the United Kingdom's Nuffield Council on Bioethics. Third will be Professor Alta Charo. Alta is currently, during the 2019 and 20 year, a Berggruen Fellow at the Center for Advanced Study of Behavioral Sciences at Stanford University. And her regular job is as Warren P. Knowles Professor of Law at the University of Wisconsin-Madison. And our last panelist will be Rob Califf. Dr. Califf is Vice Chancellor of Health Data Science for the Duke University School of Medicine, where he's also Director of Duke Ford's Duke Center for Health Data Science and the Donald F. Fortin, MD, Professor of Cardiology. He's also an advisor for Verily Life Sciences. So before I turn it over to Feng for the first talk, let me just sort of set the stage. And it probably doesn't need a whole lot of stage setting. But of course, the topic of today's meeting has been focused on technology. And over the last months and weeks, maybe even the last few years, there has been no more talked about and focused on technology in life sciences than that of genome editing and the tools like CRISPR and other. The focus of this panel will be on the use of those tools related to focus on an application to human genome editing, and as you see on the slide, navigating ethics and global governance. There are numerous efforts going on, and you heard this morning during Dr. Zhao's remarks about an ongoing set of international commissions, one co-sponsored by the national academies here, another by the World Health Organization, and there have been numerous other international efforts that predate those, going back to the 2017 report from the National Academy of Sciences and Medicine here, which was co-chaired by one of our panelists, Alta Charo. So there has been lots of focus and attention to the ethics and governance issues, still work to be done, and the focus of this panel will be to, I think, first lay out some of the issues and topics, and then I hope they will be a little bit provocative for the discussion that first we will have amongst the panelists, and then we will open it up to the full audience. For fuller biographies, of course, I refer you to the programs, and with that, I will turn it over to Feng. Run now?
Or I'll play that in just a little bit. So thank you very much for the introduction, and it's really a great pleasure to be here. So what I want to tell you a little bit about is gene editing. So one of the really exciting advances in biology recently, in the past couple of decades, has been the ever increasing number of genomes that we have been able to sequence. The first human genome was completed more than a decade ago, and now we have more than 1 million human genomes sequenced. So by comparing the DNA sequences of different individuals, especially across healthy and also groups of people who are affected by certain disease, scientists have been able to start to identify genetic differences that contribute to disease risk. And some of these genetic differences are even causal. So examples of sickle cell disease or cystic fibrosis have specific genetic mutations that definitely will cause a disease in a person. So as we start to reveal these genetic differences, and to date there are over 7,000 different genetic mutations that have been identified, a tantalizing idea is, what if we can go into our cells and be able to get rid of the disease-causing mutation and put back in the sequence that doesn't affect health? So it's this very idea that has been worked on for a long time. Multiple iterations of technologies have been developed. And one of the most recent iterations of technology is based on a bacterial immune system called CRISPR. CRISPR, in essence, is a way to go into the DNA of our cells, find a specific spot on the genome, and then trigger processes in the cell that allow us to be able to make changes to that DNA. So I have a short video clip that describes how this process works. Can you please play the video?
It's been playing in the background.
Oh, it's been playing. Oh, sorry. Okay, we'll watch it one more time. So this blue blob here is a protein called Cas9. It's from the CRISPR immune system. And this protein can be loaded with a short RNA that serves as a search string for this protein to find the right spot on the DNA. So if you introduce this into the nucleus where the human genome DNA is, this RNA and the protein complex will search along the DNA and compares the DNA with this RNA string. And if the RNA and DNA match, then this enzyme will get triggered and will go and make a double-stranded break and then trigger the cell's repair machineries to be able to make the precise change. So there are two main pathways that are involved. The first one is called non-homologous zinc joining. It simply re-glues the two different DNA ends back together. But this is an error-prone process, so you can form a small mutation that can inactivate a gene of interest. Alternatively, you can take these two broken DNA ends and provide into the cell a repair template. And so this is a piece of DNA that's been chemically synthesized. And this new piece of DNA can recombine by annealing to the two broken DNA ends and then allow you to switch in a new piece of DNA sequence. So this is how the basic CRISPR system works, and this technology has been iterating very, very rapidly. And just this morning, my colleague David Liu reported an improved version of this called prime editing, which makes it even easier to switch in specific DNA sequences. So as we make this technology more mature and more robust and efficient, then there are a lot of really exciting applications. And broadly speaking, there are two ways that this has an implication in human health. The first is using this gene editing technology for somatic DNA editing. And so this means we can deliver this into cells in our body, not cells that become our germ cells, and directly fix those disease-affected tissue. So if someone has a liver disease, we will deliver this into hepatocytes in the liver and be able to fix those cells. Or if someone has a blood disorder like sickle cell, we will take blood cells out, fix them, and then transplant back into the person. That's the first category, and it's moving very rapidly. Human trials have already started, and it's very likely that we'll even begin to get clinical data later this year, early next year. The second human health application is more ethically challenging, and this has to do with the editing of human embryos. Last November, the scientific community in the world learned about a Chinese scientist who went ahead and edited embryos to give birth to two different baby girls who had their genomic DNA modified. And so this particular instance caused an international uproar, and now is the subject of multiple international panels trying to figure out what's the right regulatory process for these different procedures. So I'm really pleased to be here, and that's the background, and I'm looking forward to engaging in an exciting panel discussion with the other experts on the panel. Thank you.
Thank you. Thank you, Feng. Our next presentation will be from Peter Mills, who will speak on the topic of pre-implantation genome editing from bioethics to geoethics.
Thanks, Jeff. Well, Feng's given us an excellent primer on the science. I want to speak a little bit more about scientists and the responsibilities of scientists and others, and in particular in relation to the second of the two applications of genome editing he mentioned. In other words, genome editing in human reproduction. I want to make 3 points, really. First one will be to clarify what is at stake when we contemplate reproductive genome editing. The second point will be really just to reflect on the significance of the event that Feng mentioned that we heard about last November in China, the birth of the twins who were edited. And then really I'd like to just finish by reflecting on the implications of that for what happens next. So I want to start with the observation that genome editing in human reproduction is not therapeutic, not in the sense, for example, that gene therapy is therapeutic, and nor is it preventative in the sense that a public health intervention might be preventative, because in the first case, it doesn't actually treat a patient who exists. And in the second case, the risk is one which is almost voluntarily undertaken. It could be avoided completely by not conceiving children. And what that points to is what I call the therapeutic fallacy. The fallacy of believing that because, for example, something involves a medical procedure, that it has a therapeutic aim. It would be a strange therapy indeed, wouldn't it, if it had to bring its patient into existence in order to be able to treat them. What it does, pre-implantation genome editing, is it offers to fulfill the reproductive preferences of prospective parents. And those preferences might include, for example, that children they have may be genetically related to both of them, and indeed that they may not have a genetic condition that any natural child of those parents would be likely to have, or indeed that they might have a trait that any natural child of those parents wouldn't have. Now, there are a range of options already available to people who, for example, want to exclude a genetic disease from their offspring, but they don't. So these are things like pre-implantation genetic diagnosis, but also the use of donated gametes and embryos. But not all of those secure this criterion of genetic relatedness. And there are some cases, allegedly, in which none of the other options available could, even in principle, secure genetic relatedness. Now, the point in saying this is not to say that genetic relatedness is not important, but just to say that it's not the same thing as treating somebody who exists who has a particular kind of disease. And the criterion that is often advanced for implementing genome editing, the criterion of unmet medical need that would have to be satisfied, seems inapplicable in this case, or at least if it's applicable, it's constructed on some unexamined assumptions about the nature of people's reproductive freedom. So the second point, the birth of the twins in China, according to the clinician, the scientist rather, who was responsible for them, that treatment was designed to produce offspring with inherent resistance to HIV. Now, it didn't, even in his claims, conform with the criteria of unmet medical need. But he wasn't trying to demonstrate, he wasn't trying to meet that criterion of unmet medical need. What he said when he justified himself was really two things. Firstly, that he was trying to demonstrate a procedure that he thought would work, and he was trying to, secondly, create a situation that wasn't able to be created in any other way, namely HIV resistance in children of parents who did not carry a resistance trait. In other words, what he was doing was acting as an innovator. He was providing a technological solution to create a hitherto unachievable situation. And you will note in this case that genetic relatedness is not really an issue because the HIV resistance couldn't easily be achieved using third-party donors. You'd have to find homozygous donors for the CCR5 variant that he was trying to use. Now, it's possible that for offspring, HIV resistance in circumstances in which people with HIV suffer huge amounts of discrimination and are highly stigmatized, could at least conceivably be as much of a benefit as the rather idiosyncratic benefit of being genetically related to one's parents. But notice what Dr. He was doing in this case was providing a technological solution to what is arguably, at least to a large extent, a social problem, the problem of discrimination against people with HIV in parts of China. But what is intuitively objectionable about those procedures, I think, is that what it demonstrates is the creation of individuals or groups with particular characteristics and required qualities. It's not this underlines the point. It's not about treating existing people, but about what kind of people will come into existence. So if it's not a medical treatment and it shouldn't be treated as a consumer good, then I think the appropriate way to handle this is in the discourse of public health and as an object, proper object of public policy. And this is important because what we're talking about here is an emerging technology. And what I think we should do is not just focus on the innovation question, the question that everybody seems to be fixated on at the moment about the first case. What are the criteria that we should apply for the first case? What we've got to ask ourselves rather is what happens if we have a demonstrated technology of demonstrated efficacy and safety? What limits then would we put on the potential uses of this? Now, I realize I've gone slightly to the bottom of time, so I'm not going to make the third point. I'll save that for the discussion because it relates to the issues of global governance. I think Alta's probably got quite a lot to say about that herself, so thank you.
Thanks, Pete. Okay, so perfect segue to Professor Charo, who will talk about challenges for transnational governance.
Yeah, exactly. It really was. Thank you very much for that. But before I talk about some of the topics you've raised, I really want to make a point that's been overlooked, I think. Most of the people here are interested in improving the health of human beings. And by far, for the moment, the best way to do that with genome editing is to focus on agricultural applications and environmental applications. We could be looking at improving genetic engineering of things like insects to stop the spread of malaria, an experiment that's already going on with a different form of the technology, the prevention of malnutrition, a kind of golden rice 2.0, or to forestall complete ecosystem failure due to climate change by editing plants so that they are more drought resistant. These things have huge governance issues embedded in them, having to do with transnational policy. An environmental experiment that's done in the United States may have implications for Mexico or Canada or even places further away. So this is a challenging area that, from my perspective, is not getting enough attention from the human health-focused community. But focusing solely on biomedical applications. International public attention really has been focused primarily on germline applications, and so I'm going to take at least half of my 4 minutes here to talk a little bit about somatic, that is, non-heritable interventions where there really is a patient who is going to be treated for the prevention or amelioration or cure of a particular disease. There are policy and governance questions here that are going to need attention. Very few of them are entirely new. They're just a more extreme version of old problems. For example, many of the somatic therapies that are being developed with genome editing involve an in vitro step, removing cells, editing them, replacing them. This is not something that is likely to be logistically or financially feasible in much of the global south. And yet some of the diseases that are the earliest targets, like sickle cell anemia, have a higher prevalence in the global south. So what is the mechanism by which we can help to incentivize investment in areas that don't have a good business model. And like I said, not a new problem. We've got a former FDA commissioner here who knows all about the ways in which we've tried to incentivize research into rare diseases or economically orphaned drugs. So we're going to see another version of that here. The cost of care in general, even in a high-resource country like the United States, is going to be extreme. Many of these new biological-focused therapies are particularly expensive, and precision medicine is very expensive. And then there's genome editing tourism. One of the things that we saw with embryonic stem cell research is that the language was hyped up enough to try to overcome the political problems associated with embryonic research. And the result then was that there was an opportunity for snake oil salesmen to come in and use that language to entice people who had devastating diseases like Parkinson's and Alzheimer's, and come in and be treated for stem cell therapy, none of which had actually had adequate proof at the research stage of working, nor was there any guarantee that the cells they were using were the cells they were claiming to use, no quality control. It started in other countries, but it was present here with hundreds of clinics around the United States opening up and advertising stem cell therapy for a list of diseases that was as long as a page. I worry that genome editing may fall into the same kind of medical tourism trap. And so, again, a governance problem of how do you control cross-border movement of clinicians to find a less regulated jurisdiction, of patients who will spend money to go anywhere. How do we avoid creating what I would like to call risk havens for these kinds of snake oil salesmen? But in the last 2 minutes on germline in particular, right, to address what Peter Mills was talking about, you know, the 2017 report, and Jeff was involved in it, and many people in the audience here, I see Keith right in the front row, were also on this committee, really broke new ground by saying that heritable editing was ethically defensible, but only subject to very strict limits and conditions. And we recognize that this was not about curing an existing person. This was about satisfying the desire to have a genetically related child without having the risk of a known high, serious mutation manifesting in the child. And we put in conditions like no good alternatives, and that would include treating somatically after birth, to repair the problem with a wild type, so that you're not talking about enhancing people into, kind of superhuman capacities. You're talking about replacing what's damaged with what will function in a way that is typical and well understood. So both for ethical and for technical reasons, a wise idea, and also long-term oversight. None of which, by the way, were conditions that were satisfied by Dr. He Jiankui. Apologies for the mispronunciation of the name. So when he made that announcement in Hong Kong, and it really sent shockwaves through the community, as Feng was saying, the organizing committee for the second summit thought hard about whether to call for a moratorium and realized that there's no way to enforce such a thing. And so instead set out a plan to create a roadmap toward responsible use of this technology. And one of the results of that is the International Commission Jeff is a member of that really is, we're hoping, going to look at what are the technical questions that have to be answered in the in vitro and animal work so that a regulator is able to assess what the risks are and what the possible benefits are based on data about off-target effects or precision or whatever else is at issue here. And then be able to turn that over to regulators so that they have the tools they need, whatever their policy is, to evaluate a proposal to move this from the laboratory to an actual pregnancy and a live birth. The WHO also has a committee on global governance, and it's looking at how we have mutual recognition of different regulatory cultures, how we create an ecosystem of governance, not just legislative or regulatory, but also the role of insurers for liability or insurers for healthcare coverage or local medical societies or journal editors in vetting for compliance with ethical norms, as well as professional codes. And in conclusion, I will say that the only thing so far that the committee has actually committed to in public is a registry of those experiments that are going on. Details are still being worked out, but we decided that good policy begins with good facts, and we didn't have any facts. So the first question is who's doing what and why are they doing it? And after that, we can talk about the wisdom of doing it. Thank you.
Thanks, Alta. The last of our panelists is Rob Califf, who will talk about regulatory and clinical practices considerations.
Well, just a little bit here to start off with. As a simple country doctor from South Carolina. I came to realize pretty quickly when I was at the helm of FDA, there are two topics which are politically incendiary. One is sex or reproduction, and the other is modifying genes. Both of them. So we're at the intersection of two things that are almost impossible to have a rational political discussion about, which when you get to regulation is really a matter of politics and science all mixed together. I learned about this, just to tell a quick story. As I was prepping for Senate confirmation, I had a nice briefing on genetically engineered salmon, but I had not been in charge in the year before of the animal or agricultural side of the FDA, the food side. They forgot to tell me they were going to approve genetically engineered salmon in the near future, and I visited Senator Murkowski and assured her that I knew about genetically engineered salmon, but nothing was in the works. Within a week, the FDA announced approval of genetically engineered salmon. I had a hold put on my nomination. I was called a liar publicly. I got used to that. But, you know, this is a case where, just as an example where, you know, this is a fish, and the public concern about contamination of natural fish with genetically engineered fish was quite profound. And of course, for years we've been living in the same situation with agriculture. And if we're going to feed the world's population at a time of climate change, I don't see how we can do it if we don't come to grips with the agricultural side of this, where it's pretty straightforward. So then we moved to somatic gene editing. And I was a little amused by my colleagues, I have to say, because over the years, I spent most of my career doing phase 3 and beyond clinical trials, where the vast majority of the time, the scientist's best idea turns out to either not work at all or to cause more harm than good. So when I hear something like fix or when proven safe and effective, I just think about, holy smokes, how are you going to fix anything in this complicated human condition? And how are you going to know when things are safe and effective? To really know, you need to evaluate outcomes over a period of time which is significant enough to understand the potential for off-target effects, which could be way downstream. And it's hard for me to imagine any animal model that could possibly tell you about human off-target effects. I think a main lesson of the last 30 years of drug development is that animal models are not human models, and sometimes they're predictive, but you don't know often for a long time after you've gotten into it. The question of when is market entry acceptable versus an experimental situation. I think we've all gotten comfortable with the idea that the more serious the disease, the less effective the treatment, the more risk people are willing to take. And particularly in America, we've said that's okay. That's why over half of drugs now are approved on accelerated pathways. But that also puts you in a situation where you don't really know what the effect is going to be. You've taken a risk, and how to handle that in a regulatory sense, I think is going to be a fascinating problem, especially if there's a catastrophic thing that happens early on, as happened with gene therapy. I'm not saying that will happen, but you know, individual events can have a big impact on public opinion, so we'd better be careful now. And then, of course, you got the question of long-term follow-up. I think the good news here is that with the methods that are being developed to use social media and other means to follow people longitudinally, it's going to be possible to keep track of people for the long term at a much lower cost. But there's a lot of work to do for all therapies, much less somatic gene editing. And then we get into the germline editing, where we're part of, there's the International Committee, the WHO Committee. We're trying to figure out how to play ball in the same court. It's not entirely clear to me, but there are some daunting issues there. The one that does bother me a lot from the regulatory perspective is who is the subject of the human experiment? Because we're starting out with a human experiment. We have a paradigm where consent is obtained or assent from the parent of a child who has a disease, but telling a child who didn't exist and wouldn't have existed, you're now part of an experiment and we're going to follow you for life, or does the child have an option when the child becomes an adult, et cetera? Daunting issues from the regulatory perspective. So I just conclude by saying there's a lot, I think, that is in common with biological therapies of many types now. We do need the medical profession to step up. I never pick on Victor, but I'm glad that Victor has taken this on because, you know, there's a lot good that's happening now with biological therapies, but the kind of charlatanism that you described, Alta, is still alive and well in the United States. And I still get calls from doctors saying, why doesn't the FDA shut this down? And my answer is, where is the medical profession? These are licensed professionals doing things that have no basis in evidence that they're good and making a lot of money doing it. And I do have a worry. We used to talk about high school seniors in Bethesda had science projects on gene editing of plants. This latest revelation today, if it's gotten so easy, how would you ever know if someone started doing this? I'll stop there.
Okay. Okay, so lots to think about. We're going to have a discussion amongst the panelists for a few minutes, about 20, and then we will open it to the audience. So be thinking about your questions. Let me start, and maybe this will be a way of connecting what you were about to say at the end of your time, Pete. And that's a general question about what reasons are good enough, quote unquote, to justify the use of these tools in humans. And you may have different answers for the somatic cell applications versus the germline modification applications, but what's good enough? So maybe we can start with Alta because we took this on as part of the report in 2017, and then maybe I'll let Pete follow because I think he wants to be a little more provocative and push that envelope. So with that.
Sure. You know, the committee, and I'm seeing more members of Barry Collar, I just noticed is sitting there, talked a long time about how to cast this, and it's exactly what you were talking about as well, is who's really the subject here. And we did have to recognize the reality that what we're doing is we're answering the desire of prospective parents to have a child that's genetically related to them without running the usual risks of disease that their particular family would run. And that had to be the benefit that offset whatever risks were going to be created. And on top of that, one has to be particularly conservative when the benefits are flowing to one set of people and the risks are going to be experienced by a different person, which is the child. Granted that the parents will also share that risk because they're going to be raising that child. And there were arguments that this is simply not an important enough goal to justify any medical risk, that people in this situation should use donor gametes, they should adopt, they should be childless. Very similar to the arguments in the 1980s around in vitro fertilization, very similar to the arguments in the 1990s around pre-implantation genetic diagnosis. And what has happened is that repeatedly we have seen that there's some variation around the globe in cultures with regard to how important it is to have a child who's genetically related to you. But at least in North America, it has seemed to be an enduring value that is dearly held, and therefore we simply accepted that as opposed to making an argument that all of American culture should change because we said so. And then we looked to see if we could narrow the circumstances under which germline would be done as much as possible, recognizing that the benefit is an awkward benefit to be talking about and not a classic one involving a single patient who bears the burden and gets the benefit at the same time.
So, okay, so maybe one more sentence about what is a good enough reason to go forward? Focus, if you'd like, on the germline modification.
We talked about diseases that are serious, that often are lethal. So we heard earlier today from Keith Waylew about Tay-Sachs disease. That would be a good example of something. Second, are there no good alternatives? Now here, one of the alternatives is using pre-implantation genetic diagnosis. There are some families for whom that's unacceptable, and we debated whether something that's culturally unacceptable, the discarding of an embryo or a selective abortion, would be a way to say that there's no good alternative, and I think we kind of wussed on that one. We also talked about making sure that it was about restoring health as opposed to the wild, as opposed to enhancement. So meant that you didn't look at diseases that people could easily live with or diseases that could be treated after birth easily enough with surgery or some other treatment. So it mostly was going to be lethal diseases. And finally, the very, very unusual situation in which you have parents who are homozygous, both are homozygous, and so the child is absolutely going to get a disease like Huntington's. There was no conclusion about whether the sheer inefficiency of pre-implantation genetic diagnosis would be a sufficient justification. That is, if people can't create enough embryos to then select among them, is that a good enough justification for editing? And at the time, I think we were probably leaning away from that, but the committee never really addressed it specifically in the recommendations.
Okay. So the bar is pretty high, is the takeaway from what Alta just gave us as a very quick and quite thorough summary of a very long report. So, Pete, I want you to maybe play the counterpoint here.
You mean you want me to set the bar low?
Well, it's up to you what the bar should be, but
okay.
I know what you're going to say, I think.
I hope you wouldn't be surprised if I said that I think the bar should be set very high as well. But I do want to, there's a paradox, and I think Robert referred to it in his remarks about if you're dealing with a novel and uncertain treatment, when is the right moment to innovate? And it's complicated in this case for the second reason that whatever consequences you result from that don't accrue to a person who's capable of consenting to the procedure, but become the conditions of existence of another person. So there are all sorts of complicated questions about how you figure in your sort of moral analysis the interests of people who don't yet exist, and there are lots of sort of slightly metaphysical arguments based on non-identity and these kinds of things about how they might matter, or how they might not matter, or how they might. So on the one hand, you might imagine that if you had a treatment that could benefit them, that there would be a positive obligation to use it. And the contrary point would be that if this is a condition of the existence of a particular person who couldn't have any other existence because it's simply baked into the fact that they are alive, then you might think, well, as long as that person has a life that's worth living. So these are the two extremes. If that person has a life worth living, then whatever you've done hasn't been done to them in that sense. I don't want to get into those kind of complications, but I do want to still separate this question of innovation from the question of what you do sort of slightly further down the line. And the question of innovation, it seems to me, is a question about how you set the standard of what is proportionate. Now, the standard of proportionality for introducing a new medicine has to be, is to do with whatever other, given that you've got a situation where people are experiencing a disease or a disability, and that there are certain treatments with known efficacies available, that becomes then your kind of standard for introducing a new one. In this case, you're not talking about that. So really what you need to set, Alta said it, what you need to set against the uncertainty of introducing the treatment is the value that you attach to the desire to have, and applying the principle of sufficient reason to this. The key thing about this, in most cases, is the desire to have a genetically related child, because in most other cases you could achieve that by other means, and that really points to a feature that I think I probably didn't get around to emphasizing sufficiently in the remarks that I made earlier about, and it's brought out in this contrast between the claims that were made by Dr. He in China versus the attitude that's been taken by a lot of the national academies and other bodies that have pronounced on this. The extent to which these values are potentially culturally relative or constructed in a social way. That they're not. It's not really about a question of estimating risk, and there are therefore all sorts of prior questions about who is entitled, for example, to attribute significance to the risk and how you give significance to the risk, even if you're in a position where you're able to estimate what the likelihood of different outcomes might be. That is very different from a situation, as I say, where you have a treatment that might appear to work. So in the case of PGD, for example, one could envisage, I don't know how far down the line, but potentially a little bit further down the line, where if genome editors are demonstrated to be effective, that they become the presumptive treatment rather than PGD, because you've got a greater likelihood in that, if they're effective, you've got a greater likelihood of securing the outcome that you're aiming for by using those treatments.
So maybe I'll ask Feng to pick up on exactly that last point. You, among the people on the stage, have the most experience by far, right, in using these tools. So maybe you can speak to this sort of, what's a good enough reason, or when is it likely to be a good enough tool that we see it as the tool of choice, effectively?
So earlier this year, me and a number of colleagues, we wrote an article calling for a global moratorium surrounding the use of this technology for human embryo editing. And I think given both where the technology is now and also where the science is now, there really isn't any good case where we want to use this technology. PGD is a very powerful alternative for screening for genetic mutations, because if one were to use gene editing to modify embryo, given the current state of the technology, we have to go through in vitro fertilization. And as soon as you in vitro fertilize the embryo, then that opens up the opportunity to do PGD without doing genome editing. And the challenge with doing genome editing is that even when you edit an embryo, there's no way that you will know for sure that every single cell within that embryo got edited. So it's very likely that the embryo will be a mosaic. So some of the cells are edited and some are not. And depending on the disease, that may or may not be sufficient to overcome the pathology. So because of all these things, there really isn't a clear justifiable reason for using gene editing in this particular context. Now, stem cell technology is moving very rapidly. Scientists are already beginning to be able to develop from skin cells induced pluripotent stem cells, and then from there try to develop germ cells, sperms and egg cells. As those technologies move forward into the future and become ready to be applied, then the society is really in a position where we have to have a good approach to think about how to use this. But until then, I think PGD and also just the astronomically rare instance of when two parents are both homozygous for a mutation that exists in 1 out of 100,000 or 1 million people, and for both of them to really fall in love and want to have a child together, I think that scenario is just very, very rare, and probably non-existent. Anyway.
Helpful. Rob, do you want to weigh in on this
A couple of
particular
quick
question?
points. Boy, in all this technical talk, it's hard to imagine love, but hopefully there will still be a place for love in all this. Secondly, I can't imagine having this conversation in a political context right now. The third thing is, for the whole spectrum of all these biological things that you just brought up, if we don't develop I'll just call it post-market system that's super great, we're going to be in big trouble, because the only thing I'm 100% sure of is that there will be surprises that were completely unexpected, some good and a lot of them not so good, that will happen at some unspecified period of time after the experiment is done. So far, that's been true 100% of the time in medical therapeutics. And then finally, Alta's call for the international registry. Right now, that would really be preclinical work. Trying to get scientists to do registries of preclinical work, so far it's 0 for 2,000, actually making that happen, but it would be a great day if it ever did, because we get rid of all these biased reporting of positive results and that are in the literature.
So I think that's a really important point. And among the things I think it would be worth saying out loud is registries like clinicaltrials.gov, which people point to as sort of the example, have also opened the way for what we would think of as snake oil salespeople of things like stem cell therapies to post their supposed trials where there isn't sufficient vetting of the things that are posted. So I think it's a double-edged sword. You got to be careful about registries, and a little bit to Rob's point.
If I may just
Sure.
just to clear, though, the registry that the WHO is talking about also addresses somatic editing, and so there would be clinical trial information on there. In fact, that's probably where it's going to be starting, because that's the easiest to get at, because your point about preclinical work is well taken.
I want to focus us for maybe the last part of our time during this part of the panel on the challenges of governance, since that's part of the title for the session, and I think really a hard nut to crack. And I'm going to start at the other end this time and ask Rob to opine. So international governance in biomedicine doesn't have great examples. We don't have a track record to point to say, do it like this. So what's the right way to think about that? Where should we focus first? What models might we look at? Where does governance sort of come from in a context like the one we're talking about?
Well, I mean, let's just be frank here. We're going in two directions globally right now: globalism and tribalism. And if we go the tribalism route, there is no solution to this problem, and every country is going to have to fend for itself. And I have no earthly idea how to do that, given the fact that science is not going to be bounded by international boundaries. If we go in a more globalist direction, there's tremendous goodwill across regulatory agencies right now. Although I'm 2.5 years out from the FDA, I've kept, I think Scott did a great job, and now we've got FDA leadership that, connects very well. I will say I had no earthly idea how complicated the intersection of the human part and the medical part and other parts of international agreements are. It's one element that people are going to have to pay attention to. There can be treaties and other agreements that completely override what makes a lot of sense from medical regulation. But, just frankly put, when we get into human studies, it should be illegal globally to do a human experiment without registering your results. And I think we need to strengthen the follow-up phase and the reporting of results, not just for the primary period, but for a long time afterward in this arena. So regardless of which party wins in the next election, this next policy window, I think people need to be poised and ready to really, in my view, push for international agreements that go beyond scientific academies talking with each other and get more to a hardcore regulatory approach.
Feng, do you have any thoughts on this?
I agree, absolutely.
Ditto.
Yeah, ditto.
Okay. Pete.
Well, had I got to my third point, I think I probably would have said that it seems to me that there are three. I mean, I'm thinking again principally about the case of pre-implantation genome editing, so genome editing of embryos, but I think this also goes for potentially for somatic treatments as well. I think there are really three crucial elements that would need to be put in place, and the first is really that the global science has, I think it was demonstrated by the case in China, has really shown what I think almost equates to what sociologist Ulrich Beck would have called organized irresponsibility. There was a kind of culture of everybody doing what they feel they should do, but the system as a whole doesn't really take charge of the consequences. So I think there's a role for elite global science to demonstrate some leadership by defining a research program that has certain priorities. But I mean, it was really noticeable to me that the first, I think, 4 papers on genome editing in human embryos were clinically translatable variants. So they were for, Feng will remind me if I'm wrong, but they were for the HPB gene, the CCR5 gene, MYPBC3. So these are all genes that are associated with diseases. When before that, there was a lot of more basic research that could be done. Kathy Nyken, in her presentation in Hong Kong in the same conference that the birth of the twins was announced, identified 3 areas that we really need to know about before we can know anything useful about this. How the cell cycle determines the mechanism, the choice of editing repair mechanism. Knowing something about the repair mechanism itself and about the segregation of chromosomes in early embryos, all those things need to be done. So that's one thing. The other 2 I'll go through quickly. I said that I think this is a matter not for institutional review boards. It's not about medical indications. I think this is matters of public policy. So I think there needs to be, in terms of indications that raise serious ethical issues, a public political process that involves a broad and inclusive societal debate. Feeding into that, I'm not saying that should deliver the answer, but it needs to feed into it. And because no country is an island, as you effectively said.
Well, we'll see this week maybe whether there might be an island created.
Coming up, yeah.
That was wishful thinking on my part. Knowledge, technologies, expertise, patience, these are all mobile across national borders. There are externalities for other countries on whatever anybody does in one place. So I think there is a need for, as you say, a kind of global institution to take care of this. And the global human rights mechanism offers that. And what you forget, I think, quite often sitting here, and I think this is probably, if I can do anything, a good reason for me to be here, is that in most of the rest of the world, or a lot of the rest of the world where this research is going on, this stuff is all banned, or at least the stuff that we're worried about is all banned. 29 European countries have signed and ratified the Oviedo Convention, which contains a clear legislative prohibition on the modification of heritable modifications. And those that haven't, like the UK, for example, have their own laws that are even more strict or even more complete than those kind of things, and this is true elsewhere. But I don't think the answer is that somebody will come along and make rules, but I think the role for these institutions is really to act as a kind of clearinghouse, or to provide a framework for negotiating tolerable ethical gradients, margins of appreciation that can exist between different states that are compatible with sort of peaceful coexistence.
Okay.
May I very quickly?
You get the last word. It's okay.
It only has 9 seconds
That's
left.
okay. We'll push a little bit. You get the last word on this question, and then we'll turn it to the audience.
I wanted to just step back a second and echo some of the work done by my colleague Gary Marchant at Arizona State University. He frequently makes the point that there's a difference between harmonization and coordination of policy. The idea that we're going to have a single policy for all nations around the world is unrealistic, because there are legitimate differences in attitudes about culture, about reproduction and sex, about the importance of genetic connection, you name it. So a different question, and one that I think is more workable, partly because of the cooperation among regulatory agencies, among other things, is to ask, how are we going to coordinate these things? So for example, if you have one country that has said that germline editing is illegal, and another country that says it is permissible, if a citizen from the first country goes to the second country to do germline editing, how will the first country where it's illegal act when these parents come back with a baby who's got an edited genome? We need to anticipate those questions, because believe it or not, today in France, people that come back after having used certain new reproductive technologies with their child find that the child is not recognized under French law because of the illegality of that procedure locally. We need to anticipate different levels of tolerance for risk and benefit, some of which may be about culture and some of which may be about the underlying populations, which could also change how risky something is or is not. It's that kind of coordination that will allow us to recognize the diversity of approaches and at the same time make it possible for us to have a set of norms about how this work can be done. And last, and very briefly, with embryonic stem cell research, we had at least one example of a country where their own researchers were at risk if they went outside the country to do embryonic cell research, and then came back. And so another issue of real importance to the scientific community and to the medical community for clinicians is, what are the limits, if any, on you if you simply go to another jurisdiction that's more permissive and then you want to move back to your own? For your own personal career, are you now at risk? That's another important thing to coordinate.
Excellent. Okay, with that, we're going to turn to audience questions or comments, and I think we want people to come to the microphones as in past sessions so that we can all hear you. So let's do that, and I guess we'll just start from here on my left.
Yeah. Jake Shurko at New York Law School. So I'm going to shift a question away from germline and back to the somatic editing process. So we live in an age today when gene therapies come on the market, they routinely command $1 million a treatment or more. Prices like these threaten to bankrupt some of the public payers we have, at least here in the United States. If CRISPR actually ends up, or the other genome editing technologies, at least one of which also has an IND filed, ends up being everything that we hope and promise for, and we're actually able to treat all 5,000 of these monogenic illnesses that we have, are we worried? Do the panelists consider this to be an ethical issue that we're bringing a technology the healthcare system that we have cannot afford?
Well, I mean, to me, the ethical issue is just negotiating reasonable prices for drugs. This is a drug we're talking about. If you modify a genome, and no one that I know, God didn't say it should cost $1 million. That's manufactured out of a sort of odd relationship that exists only in the US right now, where if you have a life-saving treatment and you have a patient who has no other options, that's not exactly a negotiation that's going to go on about what's going to happen. So I think this gets back to the issue of regulating the price of drugs or figuring out a way to bring them to a reasonable level in the US.
I think we also have to take into account that the technology and the development process will get cheaper. CRISPR and gene therapy in general are modular medicine. So what that means is if you're using CRISPR to treat a disease, to treat another disease, you simply design a different guide RNA. Now, all of the pharmaceutical development around the protein has already been established, and the manufacturing practices for that same RNA has also been established. So as the technology starts to address the long tail of genetic disease, the cost for each additional one will only be a fraction of what it took to develop the first one. And that, I think, should help quite a bit with the pricing in the long run.
You're too rational. Well, you remember, the price of a drug has nothing to do with the cost of developing it.
Sure. But
for innovator drugs.
Yes, but I think a lot of the pricing surrounding rare genetic disease, orphan disease, and the astronomical cost has to do with what the current cost is to take care of the patient for a long period of time, and then also what that patient population is. All of the cost that goes into developing a drug for a small population gets distributed among that small population of patients. But if you account for this long tail of genetic patients,
I agree with what you're saying.
by the same.
I agree with that part of it. I just think, $2.1 million for a single treatment, that doesn't have anything to do with what it costs to develop that particular treatment. It's what the market will bear.
Initially. But there will be pressure to push it down.
I think the sooner the better on the pressure.
That's right.
I think there's other factors, too, that need to be at least addressed that create this dilemma. One is that the business model for pharmaceutical development doesn't work well for these things. Where all the payoff has to do with you having an exclusive right to charge ridiculous prices for a period of time after your approval, as opposed to a different model for these, particularly for certain kinds of drugs or therapies, where really what you need is a different thing like milestone payments or investment by the government up front so that you don't have this incentive. In fact, necessity to recoup your expenses after you finally get your approval. And the second is, until we have a lot of the population in insurance programs that are long-term, that is where the same insurer can see not only the cost today, but the savings 20 years from now, you will not get a sensible response from the insurers at looking at the lifetime costs, which is a necessary calculation for any system that wants to triage based upon kind of bang for the buck. But we don't have that yet in the US.
Not to mention, these are for very small subpopulations of the of people. So it's hard to scale. So things will cost a lot for a few people because you can't sell them across millions. Okay. Other questions? I guess we're all on this side still. Please.
Hi, Susan Wolf, University of Minnesota. I'd like to go back to the question of U.S. governance. As you all know, several years ago, an Academy's committee looked at the RAC and ended up making the recommendation that this multi-decade committee that had a long track record of, among other things, evaluating genetic manipulation research in human beings should be abolished, and that instead it should be replaced by a broader committee at NIH that would look across biomedical innovation and really try to focus on those innovations where there wasn't adequate governance, although it wasn't clear how adequate it was to be evaluated. How do you think about that now? NIH has done it, as you know, and I wonder whether you think this was a case of very bad timing, given what's emerged since, and whether you are optimistic that a committee with such a broad purview, you know, across biomedical innovation, and such a powerful set of questions about how do you determine whether a biotechnology is or isn't adequately regulated, and whether there's a gap, and whether this committee should move in. Are you optimistic about this change in U.S. governance? Is it adequate, even domestically, for the challenges you've articulated?
I'm happy to, but do others want to opine?
It's easy, it's not adequate. We need to keep working on it. But, you know, I like the quote from one of the other panels that said there's no limit to better.
So I'll wade into this. So that committee on which I served, actually, the IOM committee that made that recommendation, focused on the fact that the technologies used in somatic cell gene therapy experiments in the US were well understood at that point. And so it was time to think about treating that kind of clinical trial and clinical research like every other kind of clinical trial and clinical research. It didn't need that special kind of oversight. Not to say that new, uncertain, untested technologies didn't, so that there should be a mechanism by which a new emerging technology would receive some kind of oversight. And NIH accepted that recommendation, sunsetted the RAC, and created something new called Next RAC, right? Which is a bit of a play on words, the Next RAC, or Next Track, however you like to articulate it, NEXTRAC. And the idea being, it's not clear that CRISPR-based technologies will go immediately to Next RAC, but it seems like an obvious place for that to happen. So my answer to your question is they need the right people on the committee to review the particular technology that's being brought to it. The RAC had a very specialized focus, and because it focused on the particular aspects of somatic cell gene therapy, the same people could review trials that were brought to it. Not clear how that will work with NextTrack. That's my sort of upshot. But I think it's the right kind of mechanism to take on the kinds of emerging technologies that CRISPR and other technologies represent.
You know, I would just add one quick thing. The surveillance inside the FDA on new technologies is much more extensive than people realize. You know, it was a real lesson for me to see how many people at FDA are looking at every field. And I think one critical part of this needs to be continued focus on interaction between FDA and NIH. Because once you start getting into human clinical trials, you've really got to have both in play, and you have to be preemptive and a little bit ahead of the game.
And the FDA's role did not change in that story that we just discussed. Yeah.
Hi there, Michelle Morse. I'm a general internal medicine doctor and one of the new RWJ Health Policy Fellows. My question was for Dr. Califf. You kind of alluded to the concerns, I guess, around physicians perhaps not necessarily stepping up into these conversations, or perhaps not necessarily being on an ethical or moral side of the conversation in terms of profiting from some of these therapies. I wanted to know if you could talk a little bit more about how you see that and how you see us addressing it, particularly since it's notoriously difficult to sway physicians, and we've failed many times in the past, such as white physicians taking care of Black patients in the pre-civil rights era. So what are the ways that you think that we can effectively influence physicians to make the right choices and kind of stand on the right side of these questions?
I wish I had an easy, I mean, I struggled with this a lot when I was at FDA, and I struggle now that I'm out when people complain to me, because there's a tendency for people to think if there's a problem out there, the FDA can fix it with, and I actually had a badge when I was FDA commissioner. They took it away, probably for good reason. I could walk into a place and say, FDA, you're under arrest or whatever. But it turns out this, legally, this boundary between practice of medicine and FDA regulation is a very tricky area. And for whatever reason, the clinical professions have veered away from enforcing standards in their own areas. Large dominant medical schools, large health systems, they just watch this stuff happening. And oftentimes, even inside the health systems, there's profit being made on totally non-evidence-based treatment. I'm adjunct at Duke and Stanford now, so I don't think either of those places is immune from this either. So I don't know an answer other than leadership and continued focus, because the law is pretty clear. I mean, the FDA deals with things that cross state lines and can especially step in when someone is advertising for things that are done across state lines. Things done in a medical practice are practice of medicine regulated at the state level in the U.S.
But the state medical societies have been very ineffectual in this area, and they really do have an opportunity. First, for education, second, for persuasion, and third, if necessary, for discipline. Because we saw in the stem cell area, clinic after clinic engaging in what was evident malpractice. It was clear malpractice. And you know what happened? Because the state medical societies did nothing, the real enforcers were the tort bar. And the first cases of patients who were injured were handled through medical malpractice cases. And finally, everybody noticed, oh yeah, we've got a problem here. And yet still, I don't see the state medical society stepping up to actually try to address this, either through CME or through disciplinary proceedings if necessary. And that is a place where you don't need the FDA to do it. It's pure.
Well, I do think we're going to need national academies to stay focused on this, because with all the stuff we've just talked about, it's a huge expansive area where it's unclear where the boundaries are and who will look out for the average person on the street who's subject to advertising and other offshore stuff, you name it, activities that are going on. And so I do think leadership is going to be critical here.
Someone at the mic here.
Talmadge King, UCSF, San Francisco. I may have gotten it wrong, but I think, Dr. Zhang, you in your presentation at least left me with the feeling that you thought that it was okay to genetically alter insects and plants, and that was okay. And then we talk about the human genome, well, maybe there are more issues. And I guess what I want to do is, if you did say that, I want to challenge you to tell me, to make me comfortable believing that what you just said is right, because my belief is that you have no idea. And that we are allowing something to happen that seems okay on the one side, to genetically engineer a mosquito that doesn't infect a person with malaria. But you have no idea what that's going to do to the overall system. So if you said that, if you didn't say that, I apologize. But if you did, I want you to explain it to me.
Well, thank you for asking questions. So first of all, I didn't say that I'm supportive of gene drives. So first of all, with modification of plants, we have been modifying plants, the genome plants for thousands of years. This is what agriculture, what selective breeding is all about. And that's engineering crops. And I think it's very promising to use genetic engineering in crops to develop plants that are more drought resistant, that give a higher agricultural yield. This is the way that we can feed the world and potentially solve some of the climate-related problems. With regard to engineering insects, I think that is much more complicated. Of course, we engineer insects in the laboratory. We make fruit flies and modify them to study genes. But to engineer insects on a global scale, to release gene drives into the wild, that is something that I think is much more tricky and also much more profound, and that we need to think a lot more before we move forward. And there also need to be safety mechanisms built in so that we can turn it off if we want to stop it.
Yeah, let me just second it on the plants. I mean, it's still baffling to me. I mean, the way crops have been developed and flowers for centuries and for decades now, radiating seeds to produce mutations, which is just a different way of doing the same thing. Why not do it in a scientific way where you know what you're doing? I think we still have to measure environmental impact for any of these things that we choose to do. And on the mosquito part, to me, that's a matter of large-scale studies that'll need to be done as we do it. But you know, when you look at the devastating impact of some infections, it seems like something that we ought to at least be working on. Whether doing it in large scale should be done is a matter of regulation and studies, I think. It gets really dicey when you get up into larger animals and humans.
We're down to our last 2 minutes. You want to say
well,
something quick, Pete?
I think you just offered me the segue. There was what I was going to say is there's one layer in the great chain of being that we haven't addressed, and that's actually farmed animals. And these are areas where genome editing is a really near-term application. These things are in trials at the moment, and they're comparatively little discussed. And I mean, there are lots of very interesting applications for disease resistance, for environmental tolerance, for all these kind of things, and for say the polecat is the kind of emblematic example of something that could potentially massively improve animal welfare. But when you're thinking about that, I mean, it's instructively contrasting to the way in which we think about human applications. But really, one is torn between this kind of ideal and non-ideal theory in thinking about this. So there's a sense in which you have an application that can improve animal welfare and improve the situation for everyone, and potentially also human health. On the other hand, you worry about the fact that you're actually compounding systems of animal husbandry that ultimately are...
But there is the other option. I call it meat on a stick, which that is using genetics ultimately where you don't need an animal, but you can just grow meat, and that's, as you know, in the works. What if you didn't have to slaughter animals?
Direction. Okay, I don't know how I'm going to wrap this up, so I'm not even going to try. Except to say, please join me in thanking all the panelists, and we'll move to the next panel.